Okay, so it's my great pleasure to introduce our last speaker, not least, Marina Lusic. She's a group leader at the brand new building of the Center for Integrative Infectious Diseases that is based in Heidelberg University, a beautiful university located in Germany. And I think that we're really lucky today because in the last decade, Marina has done an impressive work and has made seminal contributions to the understanding of the architecture of the nucleus and how it impacts HIV integration. So, Marina, please.
Thank you for the introduction and thank you for the invitation to be here. So the title has changed a little bit because I decided to present you only one part of the story and not the part regarding transcription. And most of my work focuses on the nucleus of infected T cells. And here, after integration into the cellular genome, the virus can either replicate, as you all know, or can go into latency. And the reason, so when I started doing my postdoc years ago with Mauro Giacca in Trieste, I actually wanted to study HIV transcription, but then it became apparent to us that latency is the actually very important point. And to understand latency, we started looking at integration. So with that, basically, we understood that there's this transition from productive to latent infection, which remains the black box of HIV life cycle. So, as I said, most of my interest resides in the nucleus, and this is just an overview of how complex mammalian nucleus is. And there are different levels of organization. So at the higher level of organization, there are chromosomal territories. And within them, there are patches of open and repressed chromatin, which have been defined more recently by the development of chromosomal conformation capturing techniques. And so we now know that there are so-called topological domains in which genes interact with each other, and these domains basically then form compartments which can be either blue or red, which means white, open chromatin, active compartments, or repressed compartments, which are here in red. And so, in particular importance are then the further interactions that are occurring in these compartments, and especially are these interactions between super enhancers. So those are regulatory regions which basically allow transcription and are known to allow transcription of cell type specific genes. So this is how it all looks in more detail. So at the top of the slide, you can see how mapping of chromosomal territories looks like. And then in the middle panel, you can see that these maps are usually obtained by fish. And then, of course, confocal microscopy and now super resolution microscopy allows us to see in many details how this all looks in the nucleus. And my lab has started exploring this because we wanted to understand where in the nucleus HIV resides. And indeed, this is also very important to address because the virus infects sufficiently activated CD4 T cells, but goes into latency in resting T cells. And these two states are very interchangeable. So the infection and integration in activated CD4 T cells is at least 10 times more efficient than in the resting ones. And when you look at the structure of the chromatin here, this also seems logical in terms that open chromatin will allow the virus to go in there, whereas the closed chromatin will not allow this to happen. And so what we wanted to understand is what features define HIV integration while at the same time allowing establishment and the exit from latency. And as I said, my lab started addressing this a couple of years back when we did the first fish experiments on infected CD4 T cells in culture, and we also did this on patient material. And what we observed by using a combination of techniques is that HIV integrates preferentially in these outer shells of the nucleus, and that it integrates in the proximity of nuclear pore compartment. And this is exactly where the transcribing virus resides. The viral genome, which is latent, can also remain in the periphery, and then it's not associating with nuclear pore proteins. So the nuclear periphery, which is shown here in these red dots, like macaronis, they represent basically a very repressed chromatin environment, which is known as lamina-associated domains, or LADs. And the interesting thing is that HIV is never found in these LADs. So after we observed the positioning of the virus and a little bit went into details of this, we actually did the first analysis of the genes into which HIV integrates, and we were interested in seeing where it recurrently integrates. So kind of definition of hotspots of viral integration. And what we did initially was to look at these different 6 lists. We had a total of 1,136 integration sites, and we came up with a list of 156 recurrent integration genes. So when we mapped these genes on human chromosomes, we could observe that they somehow cluster in certain regions of the chromosomes. I'm sorry for the resolution of this. This wasn't easy to create. But what we did was then to look by FISH, so fluorescence in situ hybridization, where these genes reside in the nucleus. And here we used now FISH against some of these genes. You can see that there are 2 dots now, and basically most of these genes also seem to be peripheral. And what we also observed is that these are active genes, of course, associated with RNA pol II and open chromatin, and are excluded from LADs, from lamina-associated domains. And this is just a summary what we found. So when we do all these FISH analyses and we look at the nucleus that can be divided in these 3 zones of equal surface, we basically see that RIGs, which are here showed with the red line, have a different disposition in primary CD4 T cells with respect to other genes which are also active but are not targeted by HIV. So this was really interesting, but at the same time, a couple of publications came out, 2 in Science, 1 in Cell, in which a large number of patient data were analyzed, and then it has been proposed that HIV integration leads to clonal expansion, and that sites of integration have something to do with it. So I will again show you this image in which we have the resting cells with condensed chromatin and activated cells with open chromatin. And so what we did was actually to think what is happening. So if these integration sites, which were defined from patients who were on therapy, actually are connected with HIV latency, so the virus is not transcribing. So the reasoning was, if we have now this virus which is not active, it's probably happening in these resting cells. Do we see differences if we compare patient data with primary CD4 infections performed in vitro, so like 3 or 4 days upon infection? And to do this, we now created a new list which contained all these patient data plus our own integration sites, and a study by Brady from Bushman in which only CD4 T cells were analyzed. And we came up with a much larger list in which we have 14,000 integrations into 11,000 genes. And as you can see, the percentage of HIV integration into genes is more than 80%. And then when we merged this together, we came up with 1,831 genes which are recurrently occurring at least in 2 lists of data sets. So what we wanted to know is now what is happening with these genes, why are they so peculiar, what are their chromatin signatures, how are they organized. And we first defined the chromatin landscape of these genes by doing a number of chromatin immunoprecipitations genome-wide. And I don't want to go into details of this, I can just show you that rigs are here in red, whereas non-rigs are in black, and so there are clearly important differences between rigs and non-rigs. What we found out is when we then did the RNA-seq on these genes and we did the gene ontology is that these genes are very important for T-cell identity. And in one of the examples, you can appreciate FOXP1 as one of the genes which is important for T cells, has certain signatures which are K27 acetylation, BRD4, and in the bottom panel you can see the red dots which represent different integration sites which are found, and they perfectly correspond to these signatures of K27 and BRD4. What does that mean at the genome level? This means that these genes are genes that have super enhancers. So these are genes which are important for T-cell identity and which actually have these regulatory elements which allow large range interactions. So as I mentioned at the beginning, they are important for this folding of the genome and for the organization in general. So when we actually observe this, we then ask whether there is a specific structure, not only chromatin signature, but the structure of the genome which is involved here. And for that, I collaborated with Guillaume Fillon from GRC in Barcelona, who did the high-C analysis on Jurkat cells. It was a very deep and detailed analysis, which actually allowed us to understand what are the contact maps between these insertion sites of HIV. And what we observed is that, so it's a little bit complicated. I'll try to make it as simple as possible. Those regions which have HIV interactions will interact much more with other regions that have HIV insertions. And so basically HIV insertions or integrations into the genome can be also the predictive force between the contacts between these two regions. And not only when HIV integrates into genes that have super enhancers, it's almost certain that this will interact with another region on a different chromosome that also has super enhancers. So this is making our understanding of HIV genome regulation even more complicated, because it's not only the HIV genome that we need to take into account, and not only the sequences which are surrounding it immediately, but also the contacts which are coming from other genes and from other chromosomes. And the details of this analysis have been actually revealed because Guillaume did a lot of ChIP-seq data analysis, and he defined basically 5 types of chromatin. So there are 2 A compartments, so compartments in which genes are expressed, and 3 compartments in which genes are not expressed mostly. HIV integrates preferentially in these positive compartments, but it integrates much more efficiently in the A1 compartment than in A2. So these are both compartments of active genes, but it goes more into A1 because this compartment has more genes with super enhancers. And we were very excited when we learned this, but then we had to do the modeling as we were asked to do so on the revision. And basically what came out from this modeling is that the predictive feature of HIV integration when it's looked at globally, so it remains, however, gene expression. So gene expression is the major predictive force when general HIV integration is taken into consideration. But when we take into consideration the hotspots of integration, so these recurrently targeted sites, which can also be selected under therapy, then these clusters of genes and contacts in 3D are the main predictive force, together with gene expression, of course, and also gene size. Super enhancers, unfortunately, do not seem to predict efficiently whether a gene will become a hotspot or not. So why are they important? And again, this slide in which we actually wanted to understand what is happening during the activation process of T cells. So whether there is something specific that is going on that can explain why HIV targets genes with super enhancers which are contacting other regions of the genome. And so what we did is to do FISH analysis on a number of genes that are HIV targets and have super enhancers. And we observed that during the activation process of T cells, these genes are repositioned in the 3D nuclear space. So they move from the central nuclear position towards the outer shells of the nucleus. And this is statistically significant for all of these genes, whereas when we looked at active genes which do not have super enhancers, these genes do not move. They do not change their position in the 3D nuclear space. And at the global level, this is how it looks. So active genes with no super enhancers shown in the middle panel in white remain mostly where they are, whereas when you look at super enhancers, genes with super enhancers, they move towards the outer shells of the nucleus. So genes with super enhancers seem to be the driving force behind the let's say, changes in the genome of T cells. And an interesting correlation also came out recently from a work that I did in collaboration with my Italian physicist, in which we compared, this is a little bit complicated, but I like to show this slide because it actually confirms that even if you do statistical modeling and quantitative comparison of the reaction diffusion model, very complex physics here, we still basically see that HIV actually integration depends a lot on the structure of the T cell nucleus. So the fact that we have the redistribution of genes during T cell activation is somehow shaping the genome for HIV integration. And last but not least, what we did in order to a little bit more understand what is happening with super enhancers, we used JT1, which is a drug that is used to reactivate HIV in some cases. And when we pre-treated T cells before activation with JT1 and then activated them, the genes which would move did not move. They stayed in the same place. So this is an indication that indeed super enhancers drive this structure of the nucleus. And to add this, I will just show you here the conclusions, but I will actually skip them and show you the model in which we propose that the reorganization of T cells during the activation process shapes the genome in such a way that some genes can be more efficiently targeted than the others. And especially these are genes that, due to the fact that they have super enhancers, are repositioned to the outer shells of the nucleus, where they interact with other active genes and create a sort of hubs of genes, most probably under the nuclear pore where HIV will integrate. And so just one thought that I have about this. This is probably very important also for the reactivation of the virus, as this association of genes with the nuclear pore component and also super enhancers are involved in the so-called phenomenon of transcriptional memory. So we are now continuing the study to understand whether different stimuli which are given to the cells that are infected can reactivate the virus in different ways. And especially this would be interesting also to see whether the priming of T cells with, for example, interleukin-7 and interleukin-15 instead of interleukin-2 and CD3, CD28 antibodies could reshape the genome in a different way. And yes, with that, I will finish. I will just show you the people who did the lab, who did the work, my lab in the top panel, the black building in the bottom, and people who did the lab, the work, the wonderful collaboration with Guillaume and his team in Barcelona, and then a group of bioinformaticians in Zagreb, and a microscopy unit in IMB Mainz headed by my friend Vasilis Roukos, and all the funding. So thank you very much for your attention.
So we have time for questions now. Pepe?
Thank you. It's really fascinating. I have two questions. One is, do you think that depending on the entry of the virus through the nuclear pore, associated or not with different proteins, I'm thinking in transporting CSF6,
Yes.
LEDGEF, the depth of the virus is different, or the integration size of the virus can be different?
I absolutely think this, and we try to look a little bit into macrophages as myeloid cells, which are also infected by HIV. Unfortunately, there is a very limited amount of integration sites in macrophages, and as compared to T cells, in which you have thousands, if not millions, in macrophages there are only 1,700. And also, there seems to be a very big difference because a nuclear pore is opening differently in T cells versus macrophages. And this is because macrophages are adherent cells, whereas T cells are not. And so the diameter of the pore seems to be different, thus allowing, for example, the capsid entry versus non-capsid. So we are interested very much in this, and I'm doing some work with Hans-Georg to understand what is happening there. And we have a wonderful collaboration with a nuclear pore group from EMBL who are real experts on it. So we hope to get some answers in that sense.
And the second question, you mentioned in your last sentence the differences between the different T-cell activation.
Yes.
So, concerning, for example, pure homeostatic proliferation with IL-2 or IL-7 versus T-cell activation through the TCR, have you some data suggesting that integration sites could be different, or these super enhancer regions also be different according to the stimuli used?
Well, so I don't have any data on integration sites, but there are, so the whole concept of super enhancers, which is relatively new, has been explored to a certain extent, and not in enough details to basically define different subsets of T cells, and especially not different stimuli, but I believe that there are differences. And the reason for that is because all these cytokines, they have different modes of action, and they also give rise to different responses. So the gene expression patterns are probably different there, and therefore I think also somehow the reshaping of the genome might be different. I'm not 100% sure, but this is my idea.
Marina, outstanding work, very nice presentation, so congratulations. My question is regarding you showed that the super enhancer region in chromatin is the key locations for the provirus that are integrated, and that this HIV integrated and somehow is a sticky or attracting force to then rearrange again chromatin approaching different genes for different chromosomes. So I understood this is the game. So Do you know, is this after integration, this chromatin enrichment directed by the integrated HIV conditions somehow the profile of gene expressions under regulation that allow these infected cells to escape the immune responses, just favoring a reservoir and latency?
No, I don't know, because what we would have done ideally was all these contacts also on infected cells. We did not do that. We are now setting up a method to actually use HIV as a bait and map these long-range interactions. It's not easy because there were some drawbacks in a sense that if we map the long-range interactions, then we won't be able to see the direct integration sites. But we came up with a possible solution, and we are now trying to set this up. So I hope that in the next year or so we will be able to answer the question whether the presence of HIV actually changes also the conformation. There has been recently a paper in Nature Communication by Christiane Nouveau from Paris, in which she did a similar work on HBV, and HBV did not change the landscape, although it does not really integrate. I mean, it does in that CCC form, which is not infectious then, but none of these forms changed the structure. Whereas they were asked during the revision to do, because she told me this, I didn't review that paper, that adenovirus infection, although it's not integrating, because the genome is bigger, it provokes chromosomal rearrangements. And so taking all this into account, I don't think that HIV alone as 10 kbs integrated into human genome will dramatically change the landscape, but it might enhance some contacts because of the LTRs and being very strong promoters, etc., etc.
Thank you.
Thank you, very, very nice. I like it a lot. We tend to think that usually when we measure reservoirs that when we measure one copy, we measure one cell infected. My question is, if this should be true? I mean, there is just one copy per cell, there is just one integration site, or is this changing depending in different genes, if the integration is in different genes, so it's replicating after first step of integration?
So this is a question of debate in the field, because if you look at in vitro infections, then you give as much virus as you want, and in dependence of the multiplicity of infection, you will have different copies. There was recently a paper in Cell Host and Microbe by Alan Engelman, in which he shows fish results with multiple copies, which to me is total nonsense. I'm sorry to say this, but if you remain at low levels of viral loads which are used, we normally see 1 or 2 copies. When we do the same experiment in macrophages, we normally see at least 1 copy more. So in macrophages, these numbers switch from 1 to 2 in T cells to 2 to 4 in macrophages. We still don't know, however, because we don't have definite proof that what we're seeing with fish is really integrated. And the reason for this is because even if you use raltegravir to block integration, you still see the signal. And this signal is diverse, it's not as strong, and usually it's not peripheral in T cells. Then the virus kind of, this was published in the Nature paper. So ALU-PCR does not tell us how many copies we have. If fish is really detecting 1 single copy, I think it is. But then some people will tell me maybe you have concatamers of HIV integrating, but I believe that somebody would have seen this so far. So yeah, that's mostly 1 to 2 to answer your question in T cells. In patients, when we did it, we usually saw only one copy.
Thank you.
We have a question in the back, Maria Jose.
Thank you. Very nice data. I have one question. Do you think your data is supporting the hypothesis that resting cells can be infected and establish latency directly without going through an activation phase?
Well, we did not do direct infection of resting T cells. So I cannot, I mean, we leave activated cells to revert back to latency. But I don't know. We tried to do, so the efficiency of resting T cell infection is much less. And so then fishing for that infected cell in fish would be very complicated. We will probably have to think about sorting the cells, which we will probably be able to do in the next couple of months. And yes, I do not exclude that resting can be infected, but I think that the efficiency is much lower.
So, thank you everyone for being here in this interesting and exhaustive session, and I think we are now all ready for lunch.
Okay.
Thank you.
Thank you.